Bonjour à tous. Je crois que nous allons commencer.
Well, hello everyone. I think we'll get started. I'm Malika Diora. I'm a physiopathologist specializing in diabetes.
Allô? Ah oui, bonjour. Moi, je suis Mila Montero Sepulveda. Moi, je suis
Hi,
nutritionniste de
I'm
base.
Mila Monteiro Sepulveda. I'm a nutritionist from
Ici en
Brazil,
France, j'ai fait aussi
and
également
I
Malika.
also
J'ai fait un
work with
doctorat
Malika.
en
I have a
physiologie
PhD in
physiopathologie,
physiology and
donc dans le domaine du
physiopathology
diabète et
in
de l'obésité,
diabetes and
et aussi un
obesity,
peu de microbiota intestinale.
and a little bit of
Donc, nous sommes tous les
microbiota
deux
as
sur la
well.
prise
So
d'une nouvelle marque
we
qui
have
s'appelle
founded
Sen
a
Food.
new
Et
brand
nous avons
called
utilisé
Sin
cette
Food,
casquette pour intervenir en tant que
but we're
scientifique. Et c'est avec plaisir que nous allons
changing
vous
our
présenter
caps
un
here
sujet
to
qui nous tient à
present
cœur,
as
qui est aussi
scientists and
d'actualité.
our
Il s'agit du
startup.
lien entre
This is current.
alimentation,
It's the connection
microbiote et
between
santé.
food, microbiota, and health.
Nous allons commencer par quelques définitions. Donc, le microbiote
So, a
intestinal,
definition: intestinal microbiota. Some of you know as the intestinal flora or the gut flora. These are microorganisms in the human intestine. So each of us, each of you out there, you have about 100,000 billion, 100 trillion bacteria in the distinct part of your intestine, i.e. the colon. So people have 90% bacteria and 10% human cells, the human body, which might be staggering there for you, but weight-wise, the bacteria in our digestive tube represent 1 to 2 kilos. So it's about the size of a human brain, and that's anything but negligible. It's important to know that intestinal transit is a major factor that makes these microbial populations much denser in the colon. So here's a gradient on our slide that shows the colonization or the development in terms of the number of these bacteria from the mouth to the anus. And here you can see at the colon there are 100 billion bacteria per gram of feces.
Donc imaginez 100 milliards de bactéries.
So 100 million bacteria in the human intestine, and these are compact with the intestinal mucous membranes. And that's a good thing because this mucous membrane, the intestinal wall, is quite vast. It's the biggest surface of the human body, actually, in contact with the external world. And if you rolled it out, the wall and all the twists and turns there, that would represent some 200 square meters, i.e. the size of a tennis court. So it's a huge surface for exchanging and dialogue between these microorganisms and our human cells. And these exchanges are what we're going to look at.
Mais avant ça, j'aimerais vous présenter un petit peu plus comment se
Now, let me give you a little idea as to how the microbiota gets established in the human body. At birth, there's an inoculation phase of this microbiota, which gets increasingly complex to be considered as a functional balance to up to the age about 2 to 3 years of age. But many factors influence the colonization of the intestine, so the bacteria of the microbiota, and that from birth. And that will be the type of birth, be it C-section or normal birth, food, be it breast milk or formula. So many things at birth that can influence the colonization of these bacteria in the newborns or other elements as well. Diversification in the eating at the weaning phase, and that's why it's really important to have diverse, varied food for the children, for the babies at weaning from breast milk and and any type of antibiotic treatment. This will impact the implementation or the colonization of these bacteria, and this will bring about harmful consequences in the stability of the microbiota at the adult age. But once it's established, we can consider that it's stable over time, but the composition can change temporarily throughout life, and this could be due to balanced, imbalanced diet, any change in diet, stress, but also following antibiotics. And then the microbiota will be, say, unbalanced, and it will come back to its initial state 1 to 2 months later. However, let me stress here that a succession of these temporary imbalances through antibiotic treatment, lots of stress, or anything else, the changes in the microbiota can have very harmful impact on health. If you look at a lawn that's been improperly mowed, you can see certain places you may have holes where there's no grass, or sometimes it's too long. So that could be the exact same thing in the intestine. Now, before I pass over to my colleague, I'd like to say a bit more about the characterization of the intestinal microbiota. So amongst the species in microbiota, most of them are strict anaerobic microbiota, with some 19%, 99% of the population being in three major families: the bacteroids, the firmicutes, and the actinobacteria. And then there's a subdominant microbiota made up of anaerobic bacteria and a passing microbiota made up of lactic and yeast bacteria. So now to understand the composition, scientists have used laboratory techniques, but most of them are in aerobic conditions, i.e. where there's oxygen present, whereas I said most of the bacteria are anaerobic. So this technique of cell cultures and microbiology have enabled us to expose or characterize only 30% of the existing bacteria in the microbiota. So this means that you need other approaches, and a recent approach based on 6S as a marker. We've determined that man has over 1,000 species, and each individual here has about normally 200 species. So we're all individual, completely different, and we have our microbiota that is specific to us in terms of the combination of the species. Now, to better understand our microbiota, in the 2000s, 2010, we had a revolution there to understand the makeup, and that's metagenomics. Now, what is that? That's a strange word there to indicate the sequencing of the genome of the bacteria in our digestive tract. And this technique of metagenomics enabled us to expose the idea of the types of bacteria. So each individual in this room could be broken up into, say, 3 types of people based on the dominant bacteria in your microbiota. So some of us have an enterotype that's bacteroid enterotype, which means that the bacteroids have come together to favor the growth of certain species, reduce other species. And many scientific studies have shown that the idea of enterotype is related to your diet over the long term. So people with the Prevotella enterotype would generally be people who have very healthy eating habits, so rich in fruits and vegetables, and so on. So as Malika explained, the types of bacteria in the intestine, I'd like to talk a little bit about the role of intestinal microbiota. You have to know that we can't live without the microbiota. They're bacteria, yes. When you're thinking bacteria, you think it's something bad, but no, this is good. It's an advantage, it's a plus in your in a healthy body to have the microbiota. So the main role is to modulate the physiology of the intestine and the development of the digestive tube. Now, this means that it participates in the formation of intestinal contractions, and that's really important. Also, in the maturation of the system, keeping in mind that the intestine can be 200 to even 400 square meters. If you roll out all of the microvelocity, it could cover 2 tennis courts. And If you look just between the enterocytes and just above the enterocytes, you have a huge quantity of cells that are they're immunitary cells that are there to defend our system, our bodies. It's the biggest surface exposed to the outside world, of course. You can imagine that when you eat, if you eat something contaminated, you have all these defense systems that come in the intestine, and you can have that can represent some 90% of the immune cells in your body. So it's quite substantial. So this bacteria is in constant dialogue across the bacteria and also with our cells and our immune cells in the intestine. So this dialogue, when things aren't going well, we're going to see here in a minute, what happens is we have the intestinal dysbiotics. So once your makeup of the microbiota has been achieved, if it's in contact with other microorganisms that we say are pathogens, the microbiota will react to defend your body. Once these come into your intestinal tube, well, they want to make sure that these cells want to make sure that the pathogens don't develop. The last function there is also nutrition, of course. Intestinal microbiota contributes to producing certain vitamins, vitamin K, certain B complex vitamins, B12, B6, for instance. So that's really important for the body, because if you don't have that bacteria, you could experience denutrition for certain micro nutrients. So the bacteria have enzymes to produce fermentation, so they can digest fibers that we can't digest. We don't have enzymes to break down the bonds necessary to digest fibers. So the bacteria can digest fibers. The metabolites of this fermentation, there are several in fact, but the most widely studied would be fatty acids, short-chain fatty acids, acetate, propionate, butyrate. But these are very important strains. Butyron substrate is the favorite for enterocytes. So acetate, for instance, the heart uses that a lot, the brain does as well. So it's very important here to think about the nutrition here, which will contribute to nourishing our body. And it can also represent energy, digestion of fiber can represent 20% of our calorie intake. So that's something that would be lost. But thanks to the microbiota, we can benefit from that extra energy. As I said, many cells, all this bacteria in our microbiota, in our intestines, and there's a dialogue. And if the dialogue doesn't go well, what happens? We have dysbiosis, and this is a change in the composition of the ecosystem in our intestines. And today we know that there's certain diseases or disorders, quite substantial, that are difficult to live with. These are due to degenerative diseases such as Parkinson. These are all related, Alzheimer's and so on. So there's a change in microbiota. In the composition, if I show you here, you see dysbiosis due to metabolic disorders, related to metabolic disorders, sorry. So dysbiosis in these, they also have cardiovascular, atherosclerosis, liver, such as liver cirrhosis of the liver, infarctus. We'll be talking more about this dysbiosis in diseases such as diabetes, and keeping in mind that diet is really important as regards the composition of the microbiota. You can make correlations here, such as cause and effect factors due to a low intake of fiber. And you can see an increase in consumption of fat as well. There's an impact. And so this creates dysbiosis in the intestinal flora. So this was published in Nature, and this is based on diversity. Malika mentioned the enterotypes. But in addition to that, we know something really important here is that there's the diversity of the microbiota. Now, what does that mean? If you think about a forest where you have trees, if your microbiota is poor in diversity, you have a forest with all of the same trees, meaning not enough diversity, not enough species of trees in the forest. So you can see that this is a European study by meta-hit in the microbiota in the obese patients. You can see these are obese compared to non-obese patients, micro-obese patients that are overweight. And you can see that from 20 to 40% of the patients have a impoverished microbiota. Now, that's really important. Why is that in terms of the diversity? Now, the lack of diversity, so this impoverishment of the microbiota, is connected to an increase in risk for disease risk. So if you have a poor diversity microbiota, you can have dyslipidemia, adiposity, visceral resistance to insulin, diabetes, more inflammation, circulation inflammation, so markers in the bloodstream. So we'll see an increase in adipose tissue. So these people, as I said, who are of the low diversity in their microbiota, in fact, they have more inflammatory diseases. So we know that all of the metabolic disorders are associated with inflammation. Obesity as well. Diabetes are inflammatory diseases. So this is really important. The diversity in the microbiota will act against this inflammation. And what can we do? In this study, they've combined diversity in microbiota with eating habits. This is interesting because you have three groups, or three clusters. One is the chips group, potatoes, sodas, sugar, saturated fats. Cluster 2 is a lot of fermented dairy products, and cluster 3 would be patients eating more fruit and vegetables, so more fiber. And we were able to look at the richness, the genetic richness of the cells, and it showed that more fiber intake, fruits and vegetables, gave you a richer diversity in the intestine. So you have a phenotype of your microbiota that is healthier. So if you eat more fruits and vegetables, so the 5 fruits and vegetables, there is a reason behind that, so you really do need to eat those. 5 fruits and vegetables a day. And you also see the importance of this diet as regards other diseases, diabetes, obesity. So the richer your diet is in different bacteria spaces, the less cholesterol you have, the less LDL, that's the bad cholesterol, right? And the fewer incidents of inflammation, fewer inflammation markers. So thinking then cluster 3, you have to know that cluster 3 is the phenotype, which is considered to be the healthiest. So now what can we do to improve our intestinal diversity? This is really important here. This is probably my key point. Today we know that your diet is really the core of this issue. There could be other approaches if you will. You could look at prebiotics, probiotics. You can exercise, that has an impact as well, but you can do anything, everything. You can do plant therapy as well, phytotherapy, all of that impacts your health. But if you have a good diet, that's not going to be enough. Maybe just for a short term, but resilience will be that you'll come back to your dysbiotic state. So keep in mind that, okay, probiotics is a good approach, but if you take probiotics and you stop and you keep eating wrong, well, it won't help. So first and foremost, you have to eat properly. So some just practical advice here, and I'll hand over to Malika. Now, as Mila said, it's important to diversify your diet so then you diversify the bacteria that you have in your intestine. So that's a diversified microbiota. So eat a varied diet. So avoid prepared dishes. We may have some enemies over here on the trade fair, but you want to be cooking at home. More home cooking gets away from trans fat, the wrong fatty acids. Eat as much fiber as possible. Amelia stressed that fiber really gets digested by the microbiota to produce to get rid of fatty acids and have a very positive effect on your health. And as we said, the 5 fruits and vegetables even more. So you want to have a root-rich diet as well. Why root? Because they're a good source of fiber. And so that's an important substrate for the bacteria in the microbiota. Also, I forgot to tell you, breast milk has over 50 prebiotics. So that phase is really important. The initial months of the infant. Breast milk, if you can do it, you definitely should breastfeed as long as possible, because it's been proven that this helps children develop their intestinal colonization with the right bacteria, because we have nutrients in the prebiotics in breast milk that helps these bacteria grow. So it's really, really important, in addition to the type, the phenotype, and the type of birth, be it C-section or natural birth, it's really important to breastfeed. As we said, fiber, there's food fiber, there's also fiber that you can add to your diet, such as cypicillium, oat bran, many other grains that you can add. You want to use those as often as possible in producing your recipes. That's what we do. Also, you have food stuff that's rich in polyphenols, fruits with a deep color, such as blueberries, cranberries, canneberge in French, and red berries. So there are prebiotics in those. So it's very important to vary that. And we're going to wrap up our presentation. I'd like to thank the organization here at the Trade Fair for inviting us to present to you. And if you have any questions, I don't know if we have time because I think we're running a bit late. But otherwise, you can come by our stand H41, stand H, exhibit H41. Please come by, and we'll be happy to answer any questions. Thank you all for listening.